welcome to IMPAKT TV. My name is Rosa Wevers, and together with Arjan Dunnewins, the director of IMPAKT, who's sitting next to me, we will be today's host of this episode. And we are both part of the editorial team together with Friso Wiersum and Michelle Franke. And each time we try to come up with a new interesting topic that's very current in society today. We've had a short summer break, but now we are back. And tonight we will talk about DNA surveillance with our very special guest, Heather Dewey-Hagborg. But before we dive into the fascinating topic of tonight, I'd like to ask Arjan to briefly say a few words about what IMPAKT is.
Yeah, welcome all viewers. For those of you not very familiar with IMPAKT, we're a festival and a center for media culture. We try to reflect on the changes that technology and our use of media bring to our societies, our lives. And we do that every year in a festival. The first next upcoming edition of IMPAKT is actually already quite soon. It will be from the 3rd to the 7th of November. Modern Love, you can read all about it on our website, impact.nl. But also we have lots of events in our center before and after the festival, sometimes related, but also very often addressing other topics. Like for instance tonight's event, but also the exhibition that is coming up and that you'll hear more about in this event, Face Value, also featuring Heather Dewey-Hagborg. So that's IMPAKT in a nutshell. We're very happy that you're here. We hope to see you more often at IMPAKT TV or at our other online and offline events.
As for today, know that you can ask questions, and I can imagine that you have a lot of them. You can ask questions either via the chat or by sending an email to questions@impact.nl. Today, we're very honored to have as our guest researcher and artist Heather Dewey-Hagborg. Heather Dewey-Hagborg is not only a transdisciplinary artist, but also an educator who's interested in art and research as critical practice. Her work includes the project Stranger Visions, in which she created portrait sculptures from analyses of genetic materials such as hairs and cigarette butts that she collected in public spaces. And we'll hear more about that soon, I expect. Heather holds a PhD in electronic arts from the Rensselaer Polytechnic Institute. She is a visiting assistant professor of interactive media at NYU Abu Dhabi, a fellow at the Sundance Institute's Art of Practice program, and Heather is affiliated with research organization Data and Society. And on top of that, she's also the co-founder and co-curator of Refresh, a politically engaged collaborative platform that is situated at the intersection of art, science, and technology. I'd like to welcome you, Heather. We're very happy to have you here as a special guest. And we've asked you to give a short presentation to your work before we dive into the discussion. So without further ado, please, Heather, take the digital floor.
Hello, thank you so much for having me and such a nice welcome and introduction. I really appreciate that. I'm Heather Dewey-Hagborg and I'll be talking with you today about my artistic practice with a focus on these issues of genetic surveillance and then thinking a little bit more broadly about surveillance towards the end. And I'll be starting by talking with you about some of the work from almost 10 years ago now, which is Stranger Visions, and then we'll end up with talking about some more recent projects from just in the last years. So, where did it begin? So how did I end up working with this stranger's DNA? So this began in about 2012, a little bit earlier, where I was sitting in a therapy session and I was staring at this print across from me on the wall, this very blasé kind of therapist office print, and noticed suddenly that the glass covering the print was cracked and there was this hair stuck in the crack of the glass. And I sat there for the hour and I just kept looking at this hair and thinking about it, thinking about the person who had left it behind, imagining them and really wondering what could I learn about them from this bit of themselves that they'd forgotten about. And when I left, it occurred to me that the very things that make us human, the messiness of the human body, was increasingly becoming a liability as we've constantly faced the possibility of shedding these kinds of traces in public and leaving artifacts which anyone could potentially come along and mine for information. And so in Stranger Visions, that's exactly what I did. I began obsessively collecting those hairs, those bits of chewed up gum, cigarette butts, nail clippings from the streets and public bathrooms and waiting rooms of New York City. I extracted DNA from them and analyzed it to generate 3D, life-size, full-color portraits, 3D printed, representing what those individuals might look like based on genomic research. And to do this, I looked at a variety of different traits. So I looked at certain things that are fairly well established, like eye color. I looked at more contentious genetic loci associated, for example, with ancestry, where the production of genomic knowledge about populations is more disputed. And I looked at genes that are not often considered controversial, but which I think do demand further deconstruction and analysis, which we'll also talk more about, like sex. And speculative science as well. So I looked at preliminary studies of things like weight and freckling and facial shape. And when I started this project, I really knew next to nothing about genomics or molecular biology. So I was really beginning this as an amateur and coming at it from kind of the hacker tradition of digital media and computer programming. And also coming at it from this angle of art as research. So I had this question in mind: how much can I learn about a stranger from a hair? And I didn't know where it would lead me or what I would find out, where that would go. And this is how I tend to work. This is how I like to work, is to start with this kind of question and see where it takes me and really use art as a way of learning about something. And so when I started the process, I had little knowledge about kind of how to answer this question. And I had very little money as well. But luckily for me, GenSpace, the world's first community biology lab, had just opened up down the street from me in Brooklyn. And so I went there, I signed up, I took a kind of crash course in biotechnology, and over the course of several weekends, we learned how to extract DNA, how to amplify it, how to analyze it. And then ultimately I ended up working under the mentorship of the scientists there on what became Stranger Visions, working kind of cigarette butt by cigarette butt, experiment one at a time, building up this knowledge and these inferences about these strangers. So this connected with earlier work that I've been doing as well in facial recognition. I think we'll talk a bit more about this later, but basically for many years before this, I've been doing work dealing with surveillance in different kinds of ways, thinking about things like the growth of cameras on the streets, thinking about warrantless wiretapping. That was an issue that was just emerging at that time, thinking about speech recognition. And so I was doing work that was digging into the code around surveillance technologies. But what I realized is that no one was talking about the issue of bodily genetic surveillance, that this was missing from the conversation. But I had this background working with facial models and digging into facial recognition. And so what I knew is that you can take a model that's used for recognizing faces and you can turn that around and turn it into a model for generating faces. And in Stranger Visions, I built on this particular project out of Basel called Morphace. And so this is a model that was designed for doing 3D facial recognition. And you can morph the model along these different kinds of axes. So I saw this and I immediately thought, well, this is a way of generating different kinds of faces. And so I took their model and I retrained it with some more data from different kinds of facial recognition sites from the US, for example, and started kind of excavating these other axes of variation within the model. And when I went into this, I didn't really know what I was going to find. So I just suspected that this was becoming possible, this technique that we call forensic DNA phenotyping, this attempt to produce a face from nothing more than a DNA sample. And because I could see what was happening with genomic research, I could see what was happening with companies like 23andMe offering these services to anyone. And I could see what had been happening with 3D facial recognition. I was kind of putting those two things together. So I was connecting the dots, and I could see that if I could think of that, certainly someone else would be thinking of this soon too, that this would be an emerging technique for criminal investigations. But I thought that was still a little ways off. And so what happened, so what I did was I would walk through the things in the wet lab, extract the DNA, sequence different pieces of the DNA, and then bring that data into a computer program that would then spit out a kind of genetic profile of the person. And then that I would line up with this morphable model and start generating different possible versions of what someone might look like based on that DNA sample. And so I would maybe generate 5, 7 different versions of what that person could look like, because there were kind of infinitely many ways that they could look, of course, but there's some that are more probable and some that are less probable. And so I would choose 5 to 7 different ones, and I would, or output 5 to 7 different ones, and then I would look for one that meant something to me, one that made me feel something. And then I would output that as the final 3D model, touch it up in modeling software, send it to the 3D printer, print that at life size in full color. And this just gives you an idea of what that looks like in an installation setting as well. And so at this point in time, this project was meant to call attention to these new ideas of genetic surveillance, to predict a kind of problematic future that I thought was coming, and confront people with this in an intense and visceral way, to implicate them, to have them walk into the gallery, think that could be me, that could be my DNA. What do I think about that? And clearly it was meant to be controversial, and it was, and that's certainly something we can talk more about. But what I didn't expect is what came next. So that really very, very soon, within a year and a half of when I started working on, started showing Stranger Visions, a company started selling a service to police that they called DNA Snapshot, claiming to create these mug shots of suspects from DNA left at crime scenes. This was the end of 2014 that they launched this. And so in 2012, when I started Stranger Visions, I expected something like this might start coming in like 5 years, but I knew that it was much too soon. I knew the science wasn't ready. After all, I'd been just generating these handfuls of different possible faces based on DNA and choosing ones that I found artistically, aesthetically appealing. Put in the hands of police, what would their criteria be? So I think if you look at the pictures that they sell, you can immediately see how incredibly generic they are. So this picture that we're looking at here, this is actually a clipping that a friend's mother sent me from her local newspaper, where they actually say, do you recognize this man, as if it would be an actual kind of police sketch. And if you look at the details here, what you'll see is that all that they really say that they know about this person is basically that it's a white man. And so it's very clear that this is a completely generic stereotype picture. And if we look at a few more of them, you can see that this is basically what they do. They have a kind of stereotype for different races, and then they generate these different versions of it and kind of claim that they're different people, but it's really all the same face over and over again. It's very repetitive. And so there's nothing in the US, there's nothing to stop a company like this. There's no regulation, there's no scientific standards in place to specify even what might count as accurate enough information in this case to actually use. So nobody's even decided what would make it okay. I mean, what would count as an accurate DNA phenotype in this case. And so I think that putting this new and incredibly ambiguous form of profiling technology in the hands of the police, who we know, in particular in the United States, have such a bad track record of racial profiling, discrimination, and outright violence, is really a questionable thing to do. And so at this point, I started thinking, how can I problematize this. How can I bring people into questioning this technology more? Because I found it to be really, really troublesome. And so I started writing about it, and I started talking about it. I published an essay on kind of deconstructing the phenotype, but I wasn't sure yet how to bring that into my artistic practice as well. And then I got an email from out of the blue in July of 2015 from Paper Magazine that they were conducting an interview with Chelsea Manning, the US whistleblower who was imprisoned for making information public, disclosing the prevalence of civilian death and torture in the Afghanistan and Iraq wars. And Chelsea Manning was arrested as Private Bradley Manning, and after her sentencing hearing announced her name change and gender transition. And at that moment, she was sentenced, put in a military prison, and then no one could visit her. No one could see her after that moment. And Paper Magazine was doing an interview with Chelsea, and they wanted a picture to accompany it, but no one could visit her, no one could photograph her. Her image had been completely suppressed. And you could actually say that since this was also the moment of her gender transition, that no one had actually seen her. And so this piece, this use of DNA technology became an opportunity to repurpose, to rework this fraught technology to give Chelsea back the public face that she had been denied. And also, because she's transgender, we could make this the focal point of the portrait, to use this as a way of highlighting reductionism in the technology itself. So the next time Chelsea was getting her haircut, she picked up some of the clippings, and then she swabbed the insides of her cheeks with a Q-tip and mailed them to me from the prison. And I extracted DNA from them, I sent it for sequencing, and then I analyzed it just like I did with Stranger Visions, but with one crucial difference, that we decided not to look at genetic sex. And instead, we decided to present two versions of her portrait. One, a gender-neutral version, which you can see on the left, and one, a female version, which is how she self-identifies. And to exhibit these side by side to specifically show how limiting this idea of biological sex really is, and how absurd it is to have some kind of stereotyped idea of what a gendered face is supposed to look like. How is this face on the right supposedly more female? But this is exactly what we see embodied within these kinds of models, these computational models of faces. And this is what you saw a couple slides earlier, where you see literally represented within the documentation of the software this kind of slider where you can go from more male to more female. But what does that actually mean? And so if you start to see these faces side by side, you can see how really limited these concepts are, I hope. And so the 3D prints here premiered at the World Economic Forum in 2016, which was a really interesting opportunity and gave Chelsea this public face that she'd been denied at one of the most elite public events of the year. So it really kind of put her, I think, in front of people who were keeping her in prison. And to make a long story short, there's a lot more to say about that, but let's fast forward to August 5th, 2017. There's Chelsea. She was freed and able to come to an exhibit that we had together of a collaborative project at Friedman Gallery in New York City, and got to see her portraits in person for the first time. And this exhibition was a kind of celebration of her release. And when we were given this opportunity to work together on this show, we wanted to take the piece one step further. So we had produced these two portraits, but now we had a whole gallery. And we started thinking, how can we scale this up? And we came up then with the idea of the piece Probably Chelsea, which is a collaboration that shows 30 different variations of Chelsea Manning's face based on the same DNA evidence. So algorithmically generated from an analysis of her DNA, and then suspended from the ceiling as a kind of crowd at an assortment of different human heights in the center of the gallery. And what we're pointing to here is the idea that genomic data can tell a multitude of different stories about who you are, about what you are, and the installation shows how many different ways that your DNA can be interpreted as data and how subjective this act of reading DNA really is. So there's so many different possible faces Chelsea's DNA could be interpreted to represent, and this installation shows just 30 of those probabilities. Finally, the form of the installation was inspired by conversations that Chelsea and I had about the limits of DNA profiling, along with the incredible mass movement that advocated for her release from prison. So this form of this kind of crowd standing there is meant to evoke this feeling of the crowd that stood with Chelsea, that advocated for her release from prison. Probably Chelsea is meant to evoke a kind of molecular solidarity to remind us that we have so much more in common genetically than difference. Each genomic variation is a piece of data, a new clue, and a different possible story. As more data is put together, some things become more probable, some less, but there's never certainty, and there's always alternate possible narratives. So probably Chelsea portrays these alternate narratives, it gives space to them, and it represents a sampling of the many, many stories that Chelsea's DNA can tell. And with that, I would love to talk to you more about all of these things. Thank you.
Thank you so much, Heather, for this wonderful introduction. I think it evokes a lot of questions. And I have to say, this is really the case. After the time I saw your work for the first time, sometimes I have these moments when I'm in a public space and I lose a hair or I lose some DNA somewhere. And I really have these moments where sometimes I think like, is somebody picking up on this and will they make a DNA profile out of me? So I think your work really has a strong effect on people, can really make them reflect more on where they leave DNA traces. What I find very interesting about your work is that it reflects on something that we tend to see as very objective, right? So DNA is something that we equate with facts, with the truth. And I think your work very much shows that this is maybe not always so straightforward as it seems, and that there are a lot of politics and power structures actually involved when it comes to DNA profiling and phenotyping. When you first started working with DNA technologies, were you surprised to find out actually about these subjectivities? And can we understand your work as a warning or perhaps even a protest against the strong belief in the objectivity of data, of DNA?
Yes, definitely. Thank you. It's really a fantastic description and interpretation. I soundly agree with everything that you've said. And I would say that in the beginning of the process, I didn't know how much subjectivity or objectivity there was. And so it really was, I really went into it with a very kind of open mind, thinking, well, let me really find out how much I can find out about a person. And it is a tricky thing because on the one hand, I mean, there are a lot of things you can find out and they are really concerning. I mean, it is really genuinely concerning that there's so many things that you can discover about a person from some bit of themselves that they really didn't think about leaving behind. And then on the other hand, there's such a chance to misconstrue or to put your own subjectivity into the interpretation of these things and kind of into framing a person as you might want to frame them for whatever reasons. And so I realized that there was this incredible kind of ambiguity there as well. And so hopefully through probably Chelsea that begins to make that increasingly clear. But it is like with so many surveillance topics, it's really a conundrum because you have on the one hand the kind of failures of the technology where you see, well, so for example, this technology is racist or it's biased in some way, it discriminates in some way, it fails, and that's a problem. And then on the other hand, you have the parts where it doesn't fail, and that's a problem too. And so we need these ways of talking about both the strengths and the weaknesses of these technologies in a complex kind of conversation.
You're mentioning racism, you're mentioning discrimination as well, and these moments of failure. Are stereotypes something that also have a role in DNA phenotyping practices?
At this point, I would say that's basically all it is. So in terms of what the police are marketing to police, what companies are marketing to police rather, there is not much else there. I mean, it's essentially taking a bunch of samples in and associating certain parts of data with certain faces. And I think if you look at what is actually being generated by this, it's really just this one company at this point. It's one company. There are other scientific researchers working on that, but generally in a different way, in let's say in a more nuanced way. But if you look at what is coming out of, in particular, this one company, it is really blatantly a set of stereotypes. And that's clearly something that needs to be problematized and needs to be, I would say, really fought against in the hands of the police, but really anywhere. I think, we have to be on guard against falling into these kind of stereotyping behaviors all over the place. And it extends beyond just phenotyping and beyond DNA profiling to a much broader set of kind of computational tools that we can see in facial recognition as well, for example.
Thank you for pointing that out. And I think that's something that some people actually might be very surprised by, because we tend to think of DNA as sort of the personification of facts or of objectivity.
Maybe responding to what you just said about this technology being problematic when it fails and problematic when it doesn't fail. How do you navigate between this one company, or if we look at any other company, the producers also wanting to make us believe that these new technologies can do so much, which is often an exaggeration, like from commercial reasons. They of course want, for instance, the police force to believe that their DNA profiling has very accurate results, and in a way you also kind of project this future where this could happen. How much of it is then really based on what is currently actually possible, and how do you navigate with this idea that also the producers themselves are often giving an exaggerated version of the current possibilities and what this technology already can do? And this, it's with artificial intelligence. You can apply this to many phenomena, technological advancements. So how do you deal with that in your own work? How much is warning people for the possibilities, and how much is about the current possibilities?
So I would say, so certainly in Stranger Visions, it was about warning about the possibilities. I saw that would be coming, and I wanted to start a conversation about it, both about the act of DNA phenotyping and how I could see that this was a new technology that was emerging, but then also about the kind of surveillance risks that I saw coming with the kind of genetic situation more generally, which is, beyond this act of profiling and predicting a face, this increasing accessibility of DNA sequencing, of our DNA, of being able to read our DNA really to anyone who might wish to do it, with the increasing accessibility of everything from DIY community spaces to online services. And so I was definitely looking at the future, and you could say speculating in Stranger Visions. And then in Probably Chelsea, it's kind of inverting that, right? It's reflecting on it. It's trying to say, well, that is one possible future, but if we look at what we actually know, it's much more of a multitude of kind of possibilities. And so I think in a way the works, and I only talked about these two here today, but I think that they do tend to build on each other and kind of reflect on each other and fill in different bits and pieces. So then after Probably Chelsea, I worked on another work that showed other kinds of risks there that we see with the commodification of biological fluids and DNA and data, and thinking more about these direct-to-consumer services and the risks that they present in this piece called T3511, which is, I think, still open actually at the new art space in Eindhoven right now.
We're getting in questions from the audience. Just maybe to repeat, people listening and viewing this broadcast, you can ask questions via questions@impact.nl, or you can use the chat function. Maybe we'll get to those questions a bit later, or I guess you have some follow-up questions already from Heather?
Yes. Heather, what I was wondering about, we've talked about the use of DNA phenotyping in a police context, but what I also see is that there's a huge business, actually growing business, of these DNA test kits that promise to share information about your heritage on the basis of your DNA. And I was wondering, how do you look at this development?
So this is something that I've written about, in particular, just looking at DNA heritage, ancestry, these concepts, in an essay that I wrote called Sci-Fi Crime Drama with a Strong Black Lead. And so that goes in detail into kind of the problematics of these things, of both the phenotyping and the ancestry things, in terms of what it means about our ideas of race and ancestry, ethnicity. And so there's several different kinds of complexities to this here. So one is about representation. So one is about who is in the database, who are you compared against, what does it mean? So how did those data that are in the database get there in the first place? Was that through a process of consent? There's quite some databases that have samples in them that were taken from people without really having a proper process of consent. For example, when we look at what's happening with various projects and taking DNA from indigenous populations, there's always a kind of question there of what is happening with consent in these situations. And then there's another kind of problematic, which is that the databases don't evenly reflect people all over the world. So the databases themselves are really very much skewed towards European populations. So most of the research that's gone into looking at ancestry has gone into looking at tiny subclades of other subclades of groups of European populations and ancestries. So you can, in Europe, get down to the most minute, kind of detailed aspects of ancestry. And then in other parts of the world, you'll have hardly any resolution at all in those things because they just haven't been studied. And so again, you have kind of it's kind of a thorny issue there because again, it's a question of, do you want to be represented in the database actually? I mean, what does that really mean? And so again, it's a complicated question of whether and how we want these technologies to exist. And then you have kind of this other level here, which is that it feeds into a problematic and kind of base desire to quantify things like ancestry or like race or like ethnicity with percentages. And I think we can really hold the platforms responsible for this. And, you know, since this is increasingly something that we're comfortable doing and actually holding platforms accountable for the horrible behaviors that they engender in people, I think this is actually something we could really say that we could do without, is putting percentages on things like race or ancestry on these kinds of sites, because it really feeds into a reductionist and problematic view of identity. And it props up the idea of race with a seeming kind of scientific accuracy that isn't really there for something that is a social construct. It appears to give it this authority of biology.
Yeah, I think actually you responded already to a lot of things that is in this audience question that I want to ask to you. I'll read it anyway, and you maybe just see what you can add to what you already said. So we received it via the chat, and it goes, looking at the exhibit of Chelsea's faces, there appears to be a lot of scope for variation. so much so that they look like they could have been derived from different DNA sets. In your experience, can you see a future where this technology gets very precise? And can you show a near life-like representation of an individual? Or do you think that there are certain environmental factors that DNA can't represent? And secondly, when you started the Stranger Visions work, did you wonder whether it was ethical to take a stranger's DNA, or did you believe that the warning should be so bold that taking a random DNA sample would hopefully show it can be done to anyone?
So the question of kind of how accurate can it ever get, this is a really good question. It's not one that I can answer with certainty, but through having many conversations with scientists about the subject, I would say that what we can say with certainty is that we're not there yet, that we're not there today, and we may never get there. We will probably never get 100% certain, or even 99%, or probably even 95% certain about what someone looks like from their DNA. We will undoubtedly get more certain than we are now. So there's no doubt in my mind that more knowledge is going to accumulate, not just through DNA, not through genetics alone, but looking at kind of a more holistic picture, looking at genetics, looking at epigenetics, doing other kinds of cellular profiling off of evidence. And when you start putting together this more comprehensive picture, there's no question that you're going to find out more and more and more about that person. But if you will ever be able to, with total certainty, put a picture forward and say, this is definitely what this person looks like, that will always be science fiction. But we will get closer to that. I think there's no doubt about that.
Yeah.
In terms of the ethics, certainly the project was meant to be ethically provocative. So there was no question in my mind when I was doing that I was doing something that would upset some people, maybe upset a lot of people. And I made the judgment call that it served the greater good to do that as a kind of, in the tradition of, you could say, white hat hacking, of showing that there is this kind of security vulnerability there by doing it.
Did you find samples that you decided not to use because of you felt it was too problematic? Did you refuse certain samples?
So there were samples I didn't use, not so much because it was problematic as because they were too gross for me. I mean, like real, like used condoms and things like that. Didn't go there. But on the other side of that, what I did decide not to do was to profile anything like disease risks, for example, from these individuals. So I decided that was something that I wasn't going to look at, even though I certainly could have. And what I hoped with that is that the face would be this kind of index that would point people towards thinking of all of these even more invasive things that could be inferred about someone. And even these like really speculative things that are kind of absurd, like that there's genetic studies about things like guessing someone's religion or guessing their politics, or of course guessing their sexuality, things like that.
You already drew a lot of connections between DNA surveillance and another form of surveillance, which is facial recognition technology. And that type of technology is something that you also address in other work, including How Do You See Me, which will actually be on show at Impact very soon in the Face Value exhibition. Heather, could you introduce that work a bit more and say something about how politics are also a topic you're interested in relation to facial recognition technology?
Yeah, absolutely. So, like I mentioned, the Stranger Visions came from this kind of legacy of my interest in surveillance technologies, thinking about facial recognition in particular. So I had done this kind of deep work. I did a master's thesis on thinking about kind of artificial creativity through the generation of faces, using facial recognition systems to generate faces. So this was like in 2007. And so that fed into Stranger Visions. And so it's something that I've been interested in for a very long time. And like maybe 3-4 years ago, I started a fellowship with AI Now in New York. So that was with, this was an organization that Kate Crawford started and Meredith Whitaker. And they were focusing specifically on the social implications of AI. And I started thinking, how can I kind of reconnect with that work? So what do I have to say, or how can I find interest again in looking at this topic that I've looked at in a very thorough and comprehensive way already? And so the project How Do You See Me came out of that research, and you can put the slides up for that. And I'll just kind of briefly explain a bit about that. So How Do You See Me is this attempt to get closer to this other that's watching us all the time. So this blanket of cameras that are always looking at us, studying us, analyzing us. We know we're constantly surrounded by these cameras. I have like cameras looking at me right now, just sitting here in my studio. And obviously, every time I post a picture to social media or accidentally appear in one taken by someone else, algorithms scrutinize my image. And I exist in systems, even in which I don't have a profile, as a data ghost. And so I'm known secondhand as this person that's pointed to by others. And so these systems know a tremendous amount about me, but what do I know about them? How Do You See Me is my way of looking back and trying to learn about how this alien intelligence that is so attentive to my every move is structured internally. And so the question that intrigued me in researching this piece is, what do these abstractions of me look like? If we were to kind of peer inside, what would be in there? How could I visually relate to what exists within the structure of the system? And so, to put it in a more technical way, what other kinds of images might have significantly similar feature vectors to my own? And so I developed a kind of evolutionary algorithm, and I targeted facial detection and facial recognition. I kind of separated these two parts of the process apart, and developed this algorithm that would evolve pictures that would kind of score higher and higher and higher as either being detected as faces, as you see here, one just was in the video, or as, in a way, being recognized as my face, meaning that an encoding of my face for a facial recognition system has a certain vector, so a certain kind of list of numbers. And so I started to also evolve images that had a similar vector of numbers. So it's a very kind of abstract and strange way of representing a face or thinking about a person. And I'll just share a couple of these images with you. So these are images that were detected as faces. And here we can see, go to the next slide. And so here we can see another process. So this is the process of looking for these kind of strange feature vectors that neighbor my own. And this is in a way even stranger, because in the previous phases, we can somehow relate to seeing something that reminds us of a face there. I'll come back to that in a minute. But here, it's really just totally bizarre. I mean, I think we can't make any sense of this, or at least I can't make any sense of how any of the things that are neighboring my encoding in this kind of facial space could be anything like a face. And I'll show you some of these closer up. So I was kind of stunned and drawn in by these images that I present as part of this, as creating this work, How Do You See Me? And then, of course, the question is really, what does it mean? So what is being recognized here? What is being detected? And so I think on the one hand, we can say when we see a picture like this as detected as a face, this definitely points towards what is inside of the system as a kind of generic face. So we can see there's a template here of what a face means to a facial recognition system, to a facial detection system, to be specific. And we can see that it's a white oval. And so that does, I think, say something very direct about the biases that are structured into these systems. And this is something that's been very much discussed and kind of widely addressed around racial discrimination and kind of racial biases within facial detection and recognition systems. And this, I didn't expect this to come out of the process that I went through, but when it did come out, I could certainly understand why it was there because of seeing these kind of fails that have traveled across, across the media, across social media, where we've very vividly seen how facial recognition and detection systems fail for people of color. So this is kind of a very blatant way of seeing that that emerged for me. And then on the other hand, we have this kind of very strange face, this feature vector that neighbors my own, and this is this kind of glimpse in the black box of the neural network that's trained to detect and extract key features from faces. And we can see something about kind of the internal representation of that. So to conclude, I think it's really interesting to think about representation in this context. So AI is built on models, and models are these very slippery constructs. So there are these reduced descriptions of complex real-world systems. They're designed to fulfill a specific function. And so a model is a kind of artifact that comes out of this process of training, where a learning algorithm's exposed to data. And one of the things that I think is important to pull out and discuss and deconstruct is that it seems within an AI that the model makes a kind of implicit claim to represent its subject. And so the facial model seems to claim to identify what a face is, a kind of essence of faceness. And it's immediately clear when we look at this that faceness, that it is not this kind of abstract essence, it's not something universal, but it's rather a stereotype. And so we have to be cautious, I think, of this tendency to see models as objective representations and rather see them as framing identity through a particular lens of experience. And then we have this other thing that's also really interesting and strange and less obvious and harder to grasp, which is this kind of multiplicity of the subject. Because the feature vector, the abstract representation, has so many neighbors that are so alien to human experience that this whole kind of concept of similarity, I think, comes into question. And I think I'll just leave it at that. Thanks.
Yeah, thanks a lot, Heather, for another very interesting presentation. Related, as Rosa already explained, to the exhibit that we'll have with your work in it, opening soon at Impact. It made me think about the art-science kind of collaborations. I mean, the subject of your works are clearly scientific. You collaborate with scientists. The presentation that you just showed also has very strong aesthetic qualities. Where do you see for yourself how you can contribute to scientific research? It's obvious that in your work you kind of want to warn for the risk how reality can be misconstrued or be misrepresented if these scientific advancements are used or interpreted in the wrong way. But I'm very curious to hear a bit more about also maybe how you could help scientists to develop their work, or otherwise how scientists help you in your artistic practice.
Mm-hmm. Yeah, so it's a multifold relationship, definitely. I would say I have lots of different kinds of relationships to scientists. And so the different works pose different questions or challenges and gain different reactions and lead to different conversations with scientists. But one of the things that I've loved really since working on Stranger Visions is getting to enter into those scientific spaces, which means it might mean literally being invited into a lab to do a project, but it also means speaking, for example, at conferences and things like that, or having work in scientific museums, entering into this kind of space of scientific knowledge construction. So that's something that I find really, really interesting. And one of the accomplishments that I'm most proud of, actually, is that the installation Probably Chelsea was acquired by a children's science museum in San Francisco, and it sits at the entrance to the biology section. So it's really like these kids' first introduction to thinking about genetics and what does genetics mean is looking at these 30 different interpretations of Chelsea Manning's DNA. So for me, that's really like the best thing that could possibly happen in my work, is really this intervention in this area where people are first exposed to thinking about a topic. And so that's kind of one aspect of it. And then the other aspect is, I mean, another thing that's really interesting is when you do work with scientists in the lab and you really collaborate together and have these complex conversations, political conversations, that it is, again, it's almost like a performance in a way. I mean, you're bringing art into the lab. So your whole act of being there becomes also a work of art, because you're exposing people who would never normally see most artwork to this challenging form of artwork, to this artwork that's directly related to their everyday work. And suddenly they can see what they do in new ways. I mean, best case scenario, and I have had this happen in the past, where you really go in and you have this long-term relationship and they come out the other end and they say, I never thought about my work that way before. And that's, again, I think, kind of one of these best case scenarios, things that come out of it in terms of impact. And then in terms of for me, I mean, I also get a ton out of it. So for me, you know, in a way, the work that I do is always about learning. I don't think I could work on a project if I wasn't kind of fascinated by some kind of question. And so I get a ton out of it as well, because I get to learn and see kind of what does the everyday life look like of the scientists working in virology, for example, or in genomic analysis, and see, like, how do they see their work? How does that interplay with my impressions? How does that challenge what I thought about this field before? So it just makes the whole thing much more interesting for me as well.
That sounds very positive. At the other hand, I can also imagine that some artists or some scientists might be quite scared, perhaps even, or show some resistance, because your work does take quite a speculative approach sometimes. Have you encountered much resistance from the scientific community?
You know, I can't say I've encountered so much resistance. I mean, there is always skepticism. I think that is a good thing. You know, scientists are naturally skeptics. And so they will, for example, with Stranger Visions, this is a conversation I've had so many times where the scientists will come and we really get into it. You know, if like, well, what is really possible? And did you know, have a blind sample and these kinds of things. So this is usually, in my experience, how scientists approach work like that. Because it kind of, in a way, speaks their language. Now, the other experience that I've had is not getting to the scientists. So what I have found is that sometimes I want to work with scientists or with an area of research, and I can't get, for example, past the PR people. So that's, in a way, I think the PR people are much more scared than the scientists. They don't think that the public can handle knowing what's really going on. And in my experience, mostly scientists were much more open to those things. They knew, you know, that there was, I mean, if there's really ethical issues there, like around genetic engineering, for example, they know that people are concerned about these things. I mean, they aren't cut off from these conversations. And usually they want to talk about it and they want to share their opinion. and they aren't always allowed to, because they might work places where the company doesn't want them to talk about it. So that's the experience I've had most recently that's been the most disappointing, is really not being even allowed to meet scientists working in a certain place because I wasn't invited in, so to say.
Thanks. Let's take another audience question, and this is one all the way from New Zealand, which is probably not as far away for you as it is for us, but we'll get to that later. This is from Heather, currently doing a PhD in New Zealand in DNA surveillance, using lab waste, to be precise, Illumina flow cells, with Dr. Craig Hilton being one of her mentors. And her question is, I'm also interested in how you navigated the ethics in stranger vision, working with institutions, in your case the gallery. Did you construct a case argument towards the public good? That's Heather Zelmer from New Zealand.
No, Johanna Zelmer. Johanna Zelmer.
Johanna, yeah, sorry.
Yeah, wow, that's really so interesting that you're doing your studies in DNA surveillance. So that's exciting to hear about. There was no specific. It was not as orderly, I think, as your question makes it seem. It was an experimental process. So I was kind of, going through it one day at a time. I didn't know what I was going to find. I started the project with an idea, but I didn't know if I was going to get there. And really, I spent months of getting nowhere. So I was in the lab, I was trying to extract DNA, it wasn't working. I didn't know what I'd end up with at the end of the project. And so what happened was that I exhibited the work as a work in progress in a space that I was doing a residency in New York City called Clock Tower. And the Wall Street Journal came to this exhibition, and suddenly they took a bunch of pictures and put me on the front of the metro section. And from that point on, it was like a kind of media avalanche. And in a way, it in some ways kind of left my control. So there was this point at which I suddenly felt like, okay, this was not done. This project wasn't finished, but I can't keep going with it for two reasons, really. One, because it was already out there. It was on basically every news outlet around the world. And so they had kind of completed the work, and in a way that was kind of the perfect completion for the work, because it was about provoking public interest and getting a conversation going about things and so on. And so it was kind of this perfect ending to the piece that actually it was never finished, but the media finished it.
Thanks. Another question I have, you're in Abu Dhabi now, maybe you tell a bit about, but it's also something that also Rose and I pre-discussed, is the question how much the critique or reluctance or the willingness to embrace these kind of new technologies, whether it's about facial recognition or DNA surveillance, how much that is different in various cultures. We could think of maybe how many people in the US are maybe obsessed or very interested in the ancestry, often having, of course, roots in Europe, Asia, elsewhere. How do you see the differences, the cultural differences in these scientific terrains in various cultures over the globe?
Yeah, so I can of course only really speak to my experience, which is first of all coming from the United States, spending quite a bit of time in Europe, and now living here in the Middle East. And so I do feel, definitely that these things are very different point to point, even within Europe. That's very clear to me. So there's a moment in the film I made, T3511, where I'm standing in the National Biobank of Denmark. And I'm standing there in this giant robotic freezer with blood spots taken from every baby born since 1982. And it's like, it's this tremendous feeling, I think, of responsibility and trust that you feel when you're standing in a space like that. And you see that this is a very specific kind of community of people who have that level of trust in their government, that they trust them to have this totally orderly system of blood spots that are more or less connected with their national health records and all the rest. And so it's a very comprehensive system of biosurveillance, really, that's tied to these individuals. But it speaks to how those individuals see their government and see each other and trust one another. And then if you just go over to Germany, you see kind of a totally different system, which is, of course, a country that is still actively engaging with the legacy of eugenics and the horrors of World War II, and doing so many things to prevent those things from happening again. And so you can't imagine that you could have that same kind of trust in science and trust in this accumulation of records and these biological forms of data in Germany as you have in Denmark. And so that's just, for me, that kind of really highlights these dramatic differences that you can have even in two countries that are very close together.
Thank you so much, Heather. I think that leaves us with still a lot of questions and things to discuss and think about further. For those who would like to ask more questions, know that soon, at the end of September, September 27, there will be a panel next to the Face Value exhibition, where Heather also will be one of the speakers, and where some of the questions that we picked up on today will be continued there. This brings us at the end of the first IMPAKT TV episode after the summer, I should say. Thank you so much, Heather, for being here with us online. I really enjoyed our conversation. And for those who
Fantastic.
For those who are watching, know that you can still talk to other audience members at the rooftop bar after this live stream is over. And I also would like to introduce already the next episode of IMPAKT TV, which will be on music and algorithms. Thank you so much and good night.
Thanks, Heather.
Oh, thank you. That was really wonderful. Have a great evening.